Okay, hi everyone. I want to do this part two of this two-part video about energy and enzymes. We've part one talked about energy and energy transformation and the role of energy plays in metabolism. So this time we're going to start talking about enzymes and enzyme functions. Just want to point out a basic truth about chemical reactions, and that is that if you have a reaction that runs from reactants to products, they're going to have a change in delta G value usually. This one, for example, will have a reactants with higher Gibbs free energy state than the products. This is going to have a delta G value that's less than 0, which can be negative. So this reaction is spontaneous. It'll run without any energy input, right? That doesn't mean the reaction will run automatically all by itself. If you, for example, if you take ATP, which is very exergonic, minus 7.3 kcals per mole, and you put into water, it will not hydrolyze on its own automatically, quickly. It takes a while. And that's largely because you need to actually go through what's called a transition state. You need to break bonds between atoms or molecules, right? And then reform new ones to make the reaction products, right? So to do that, you need energy inputs. You got to distort bonds and you got to put atoms in the right place and so on, and then break things and reform things. And doing that requires some energy input, right? This is called the energy of activation, or EA, right? So all reactions have an energy of activation of some sort. And ATP hydrolysis requires an energy of activation. The water molecules to hydrolyze ATP to ADP and phosphate have to have a certain energy state. If they don't, the reaction is not going to happen, right? So energy of activation protects your keeps your reactions from running on their own without the presence of an assistant of some sort, right? So let's talk about this in a second. So you guys remember from chemistry that you can have molecules or crystals or arrays or whatever that are capable of lowering the energy of activation of a reaction, right? So the reaction would normally have an energy of activation like this, and if you put in a catalyst, all a catalyst does is change the energy of activation so that it's much lower without actually changing the catalyst itself. So now this is quite a bit lower energy of activation. When you run this reaction, it'll run much faster in the presence of a catalyst than without it, right? So this is incredibly important in biology. This is like a really key, right? Because in biology, you can't just heat cells up to give energy into a system to make chemical reactions run faster, right? Because that's going to damage other components of the cell. You got to keep the temperature relatively constant. Enzymes allow you to modify this energy of activation so you don't need to heat things up so much to allow a reaction to happen fast. And you guys will explore this a little bit when you do the biofuels lab number 2 this week, right? But the point is that catalysts, enzymes, lower this energy of activation, right? So all an enzyme is a protein that can do this process, lowering energy of activation. Each enzyme has its own reaction that's specific to. So most enzymes cannot catalyze any other reaction except for one, which they are able to do because of their structure, right? So you have one catalyst for just about every metabolic reaction that is going on in your cells, right? Each of them encoded into a protein, which is encoded in your genes, right? So you have thousands of these different enzymes, right? The delta G value of this reaction is not changed as a result of the catalyst, because catalyst only changes the energy of activation required, lowering it, but not the energy of the reactants or the energy of the products. So the delta G value is unaffected. It's just this hump that's lowered as a result of enzyme function, right? Should be aware, I'm not going to ask you to reproduce this anyway, but you should be aware that most enzymes carry out a reaction. They change, there's a lot of intermediate steps in a reaction that catalyst can catalyze. So you can imagine an enzyme with a substrate, that's E plus S here, goes through an energy of activation here. And in the top of that, you need this kind of state up here to make the energy, to make this reaction happen. And then you get products, okay? Actually, this would probably be a reaction without a catalyst, so no E here, just substrate going to product, right? But an enzyme will often do this by changing the intermediates in the reaction so that they're all lower energy state. So the one intermediate reaction that happens would be this one. So the energy of activation is changed from this distance, from the red line to the x-axis here, to this distance, the highest energy state, blue intermediate state energy to this place here, right? So again, this is how catalysts are working. It ends up changing, you get all these different varieties of intermediates in a reaction. Now, we don't need to worry about this. Just be aware that it's actually happening, that enzymes can do this because they can change the intermediate state of substrate being converted into product. Whereas an uncatalyzed reaction has just one way of doing this generally, right? And the molecules have to hit just right to make that happen. An enzyme allows you to modify that a little, make it happen faster. Okay, so what's happening here? There's this thing called the catalytic cycle of an enzyme. Here we have an enzyme. So here's our enzyme. It's a big tertiary structure, let's say, protein. It's a primary structure of amino acids all in a sequence, formed, linked together by peptide bonds, right? They have a primary structure that folds into secondary structures, which will be in here somewhere, and then the tertiary structure, the overall structure of this protein. There is a space in this protein called the active site, which can interact with substrate, right? And that's going to have R groups from amino acids sticking out here, which will make hydrogen bonds, ionic bonds, covalent bonds, sometimes covalent, let's start again. Hydrogen bonds, ionic bonds, and hydrophobic interactions, and occasionally covalent bonds, right? That will form in this space, right? And let's say here come our substrates, this green thing in this orange shape, and they come in and they interact with the R groups in a particular pattern to fill the active site, right? So the enzyme kind of fits these reactants into the active site, right? They embrace it. It's called induced fit. They come in, they interact, they form all these bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions generally, right? And the enzyme kind of moves in and kind of embraces it, the substrate, in such a way to make a nice tight fit, right? Again, here the substrate's held in place by weak bonds, we talked about, right? And then the active site will now have things in position to allow these two molecules to interact in some sort of way to allow them to react, right? So that the active site lowers the energy of activation and speeds up the reaction, right? By doing all sorts of different things. You look at these, right? It's a template for substrate orientation, you stress the substrates and stabilize the transition state. There's all sorts of things you can do here, right? These react to make the yellow now and blue structures, right? Those are products. And the active site no longer likes those because they're not the right shape or configuration of atoms to fit in the original active site. So they get spit out. Those are products. And the enzyme refolds back into its original shape and is ready to do this again. So this enzyme just goes through this cycle over and over again, as long as there's substrate around to react with, right? So pretty simple. Take substrate, convert into product. That's all you're doing here, but you have this intermediate, which allows the substrate to interact much more efficiently and with less energy input to make product in the presence of the enzyme or a catalyst, right? All this is a catalyst. All right, so here's an example of one. You can look at this, look at the PowerPoint when you get a chance. So here's an active site in an enzyme called alkaline phosphatase, which has here's the protein, overall structure of the protein. This is just a density map. So this is showing what this protein would actually look like if you could see it. If you could actually see the surfaces of all the atoms in the structures, what it would look like. And then inside there's this cleft here with an active site. And the blue here is the binding site that interacts with substrate, which is shown here in black. And then the catalytic site's down here, right? And this enzyme simply, I think this is alkaline phosphate. This enzyme simply is removing phosphate from things. So it hydrolyzes phosphates from larger organic molecules. So to do that, it has R groups in these amino acids in the R area, in the red area, that will interact with this and help bring water in to hydrolyze that phosphate, right? So this is basically basic scheme, right? Now, all enzymes need, not all enzymes, some enzymes need additional atoms or molecules to do this. These are called cofactors or coenzymes, right? If you need an atom to help this process go along, These are usually generally charged ions, sodium, potassium, magnesium, zinc, right? That is what they do. They interact with an active site and they sit there and they help form bonds or help break bonds or help bring another molecule in to help form or break bonds, right? As a result of there being presence in the active site. If the enzyme doesn't have the cofactor present, the enzyme doesn't function properly, right? So these are required. And this is partly why you need all these ions in your diet, right? You need a certain number of minerals. These are all minerals, right? And these are why they're important generally, right? You can't do without zinc, for example. You need zinc in your diet somewhere. If you don't have zinc, a lot of your enzymes won't function properly, right? Same with magnesium and manganese and all sorts of different things, right? Then there's coenzymes. Coenzymes are organic molecules that do the same thing. So they are molecules that are smaller and they fit in with an active site and they allow that active site to do some sort of function. Vitamins are coenzymes. So vitamin A, folic acid, vitamin B1s, vitamin C, blah, blah, blah. All these are basically coenzymes and enzymes that do certain functions. And you need these. If you don't have them, you don't do well. So this is why you need vitamins. Most of the time in humans, we don't synthesize most of these. Other organisms have to synthesize these for us, and then we eat them, consume them, and then we use them in our enzymes, right? Those are called coenzymes. All right. So there's lots of videos of this happening, and you should watch some of these. How they work, I'm not going to show them here because I don't reproduce all that well, but you should watch these. Actually, these are YouTube videos. Well, actually, some of these aren't going to work if you watch, but well, actually, I'll just run this one. Let's see if we can get this to work. Okay. All right. So, let's see, here we go. All right. Let's make this a little bigger. Yeah. Sorry. All right. So what is this? So let's try this again. All right. So here we have an enzyme, whatever this enzyme is. It has an active site marked by the green dots here. And this is where the substrate is going to come in. It's going to cause the protein to change shape. Here comes the substrate. There it is, yellow molecules. In it comes, and it binds, and the molecule shapes, and then it cleaves this. It's called hydrolysis reaction, and results in product being released. So do it again. Here it comes, right? Multiple parts of the protein is called domains. Just segments of the protein have different part structures, right? Substrate's going to come in. Here it comes, interact the active site. There we go. Here comes active site, interact, cleave, products, right? So the protein kind of goes chunk and cleaves that. All right. So cool. All right. And there's some other videos you can watch. This one's online. I think these are all available. I hope. Should probably check these links. Okay. There's a great video here called How Enzymes Work. You should watch on YouTube. That is, well, this is not on YouTube, but it's actually a PDB, but it's still, watch this. This is worthwhile. I want to watch this and show this in class if I can get to it. Right. It just shows basically how enzymes work in a generic way. And that's what you need to understand here. All right. Now, you should be aware that enzymes are influenced by different things that cause them to function ideally. They have ideal temperatures that they work at and ideal pHs and salt conditions and so on. These are called homeostatic set points. These are the various conditions that a cell maintains for optimal enzyme function, right? So if you think about this for a second, humans tend to have a body temperature of 37 degrees. We've evolved enzymes that function optimally at that temperature, which is not a big surprise, right? But if you take human enzymes and test them for function at higher temperatures or lower temperatures, they tend not to work as well, right? And you can imagine what happens at higher temperatures. The enzyme generally fall apart, they don't fold properly, and they don't work right because of denaturation. At colder temperatures, maybe the reactions, there's not enough energy in the reaction, in the product, the substrate to cause a reaction or whatever it might be, right? So there's always an optimal energy state temperature for enzyme to function. An organism that has evolved at a higher temperature like for example, thermophilic bacteria that live in high temperature water, like 79 or 75 degree water here, they have an optimal temperature profile for their enzymes. You get too hot, again, they denature, too cool, they don't run right, right? They work really well at like 70 degrees. This enzyme even does the same function. Put it at 37 degrees, may not function at all, right? Human enzymes that may do the same function as this one, you put it at 70 degrees, it's not going to function at all either, right? They've evolved to be optimize that temperature. Now you should be thinking about how that happens actually. How the protein structure in this enzyme different than in this enzyme, even though they may carry out the same function, right? So think about that a little bit. All right. Also, pH is really important. So some enzymes have evolved to be optimally functional in certain pHs. So for example, enzymes in stomach, pepsin, for example, which is an enzyme that breaks down peptide bonds, optimal pH is around 2, because that's the pH of the stomach. If you put pepsin at different pH 7 or 8, doesn't work, right? And this is the pH of blood, right? Only function at 2. So it's optimal at that temperature, at that pH. Enzymes, another enzyme that actually catalyzes, breaks peptide bonds, trypsin, actually functions in the intestines, which is not pH 2, it's more like pH 7 to 8, 7.5 to 8, right? So its optimal functionality is at pH 8, and it does not work at pH 2. Maybe it denatures, maybe there's some other problems. Be aware of that. Enzymes can be influenced by their external environment quite a bit. All right, then there's another concept here of inhibition. You can take an enzyme and it will have an active site which will carry out a reaction. So here we have one, here's our substrate, an active site interacts and will cause and make a product of some sort, right? Lowering the energy of activation of this reaction. What happens if you put a competitive a molecule in here, interacts with the active site and blocks the ability of the substrate to interact? That's called a competitive inhibitor. So a competitive inhibitor is a molecule that mimics a substrate in some way. It interacts with the active site and blocks the ability of the active site to interact with the substrate, right? Literally what's happened, this molecule is in the way. It's in the way, right? And this substrate can't get in here and form that's not, it's hydrogen bonds or ionic bonds or hydrophobic interactions to interact with the active site. This inhibitor is in the way, right? Okay, this is called a competitive inhibitor. The inhibitor competes with the substrate for access to the active site. So this will block the ability of the substrate to access the active site, right? And this will slow down the reaction, right? If you remove the competitive inhibitor, the enzyme will start to run again because the substrate will be able to interact again, right? Okay, so that's competitive inhibition. If you can also have the state where you can have what's called a non-competitive inhibitor. You can have an inhibitor of small molecule interacts with another part of the enzyme, but that doesn't interact directly with the active site. It interacts at a third or second location somewhere away from the active site. And when it does, it changes the overall 3D shape of the protein so the active site no longer will interact with the substrate. So a non-competitive inhibitor doesn't directly block access to the substrate, to the active site, but instead causes the protein to change its shape so that it can no longer interact with the substrate, right? A non-competitive inhibitor. This will also block the ability of the enzyme to function, but it is not direct. And cells use this a lot to regulate the speed of enzymes. So if you have small molecules that block enzyme function, you produce a lot of them, you can slow down the reaction here quite a bit. Substrate can't react anymore, right? But then you remove this through some sort of metabolic process, and now the enzyme will start functioning. So you can turn this on or off and turn this on or off as a result. You do that a little bit with this too, but it's not as easy. Most drugs that we are familiar with out in the world tend to be competitive inhibitors. They're small molecules and they interact with enzymes in some way to block the ability of that enzyme to function, often in the active site. Some actually are non-competitive inhibitors too, right? So be aware of that. This is where drugs come from, drugs for medicine, right? Are really important, both these types, for blocking enzyme function. Okay, there's a lot of videos of this happening. I'm not going to show you these. You can watch these on your own, right? You should be aware of this term of allosteric interactions, right? These are factors that bind to an enzyme away from the active site, like a non-competitive inhibitor. They're going to impact the ability of the active site to function, right, by changing the shape of the protein. So a non-competitive inhibitor is an allosteric inhibitor. It's also an allosteric interaction. But sometimes you can have a small molecule that interacts in a positive way. So it actually interacts with the enzyme and causes the enzyme to function better, right, to turn it on, right? So you can imagine you evolve systems where you have small molecules that turn enzymes on, some small molecules turn enzymes off. You regulate the amount of small molecule and you can regulate the function of an enzyme in a pathway, right? So allosteric interactions are often used to regulate long enzyme pathways, right? If you think about this logically, allosteric activators, the ones that turn enzymes on, are often substrates of an important process. So if you have a process that converts a substrate into a really important product, you may want to make that product a lot. If you have a lot of substrate around, it probably suggests that you need to make more of the product. You have all the substrate around. That substrate will activate its own catalyst to convert it into product, right? Or at least start the process. That's called an allosteric activator. Allosteric inhibitors are often products of a process. So you have a long path of reactions, and then you can end up with an end product, and then the product will feed back to the beginning of that long pathway of reactions and block the function of the first or an early enzyme in that process as an allosteric inhibitor, right? So this product, if you have too much product, it can feed back and shut down the production of the product. Pretty smart, right? That's how you regulate pathways, right? So you also find allosteric enzymes that respond to allosteric activators and inhibitors at the beginning of long multi-enzyme metabolic pathways, right? So you can slow or regulate the speed of a reaction, slow it or speed it up, depending upon what's happening, right? This process of doing that, when you inhibit a reaction by feedback, is feedback inhibition. When you take an allosteric enzyme and take the product of the reaction that's involved in the pathway it's involved in, and feed, and that product goes back and inhibits the function of that allosteric enzyme, that's a feedback inhibition, right? A little bit like think about how a thermostat works, right? Thermostat detects changes in temperature, and you have a temperature set point. Temperature gets too high, the thermostat feeds back to the heating system to shut it down, to keep it from producing more heat, so that the space can cool, right? Then once it cools too much, then there's a feedback activation that tells the thermostat to turn on the heating system, and it turns on, and the heating system warms the place up. Once it warms up too much, the excess heat turns off the thermostat, it shuts down the heating system, and it cools off, and so on. You do this over and over again, right? And maintain basically a constant temperature, right? So feedback inhibition is a product of a process, inhibits the process, right? Okay, now you can also have allosteric regulation of an enzyme complex. I don't think we're too much about this, just be aware of it. You can have a quaternary structure of let's say four identical enzymes all doing the same reaction, all interact with each other. You have an activator comes along, an allosteric activator, and doesn't bind at the active site, but binds in between somewhere else, and it stabilizes all these enzymes that are in the active form. So now all these enzymes are now super active, and so they'll convert a lot of substrate towards product, right? This enzymes complex can also become inactive as a result of just the whole protein structure just sort of changing shape spontaneously. An inhibitor can stabilize the inactive form, right? So it comes along, interacts in the inactive form, and blocks it, so it's stuck in that form, right? Allosteric inhibitor, stabilizing active form, right? Okay, substrate can do this too, right? Substrate can convert an inactive form of an enzyme into an active form, like we talked about, and that'd be an allosteric activator, right? Substrate, but doing this directly through the active site. Okay, so here's what feedback inhibition might look like, right? So you have this process. We're going to take the initial substrate. In this case, we're going to make isoleucine from threonine. Those are amino acids. Here's threonine, right? And we're going to have an active site of an enzyme that interacts with it. It's going to do a reaction to deaminate it. And then another enzyme is going to do something to make this series of intermediates, A to B, B to C, C to D, and then D to the final product, which is isoleucine. Now, the way this is actually evolved is that isoleucine can feedback with this first enzyme here, enzyme 1, threonine deaminase, interact with an allosteric site, right, to now force the enzyme into an inactive form. So it shuts off this. So this process no longer goes forward, right? And you don't make any more isoleucine, right? If you use the isoleucine up, so the isoleucine concentration goes down, the isoleucine may leave this enzyme and go off to be used for other processes, and this enzyme will change back to its active site, interact with threonine to now start the and you start making isoleucine again, right? So you do this, you can regulate how much isoleucine is made, keep it as constant supply. Feedback inhibition, right? Many drugs and chemicals do this. Glyphosate is one that blocks function of the production of certain amino acids, phenylalanine, tyrosine, tryptophan, right? You use a shikemic acid as a substrate, and you modify that, and this enzyme EPSP synthase is blocked by glyphosate. And what it does is it interacts with this enzyme and keeps it from making product, which should go to this next enzyme to be turned into these amino acids, right? Glyphosate blocks that. I don't know if it's a competitive inhibitor, non-competitive inhibitor, but it blocks the function of this enzyme and causes these amino acid concentrations to decrease, right? And this kills a cell. So the way glyphosate works is it starves the host, usually plants that are affected by Roundup, of phenylalanine, tyrosine, and tryptophan, and they just can't make proteins anymore because they don't have these amino acids available, right? And so they die. Done. So glyphosate is a good inhibitor of this enzyme that carries out EPSP synthase. All right, now you can do this in long pathways too. Here we have an example, ribose 5-phosphate. This is a structure that is going to be used to make nucleotides. GMP and AMP, these are monophosphate forms of nucleotides. AMP can have a phosphate added to make ADP, which can be added to have a phosphate added to make ATP, right? So that's what this refers to, that base molecule AMP. Well, to synthesize this, you need to go through many intermediates, right? You start with ribose 5-phosphate and you make this thing called PRPP, and then that gets made in this. And then there's a branch point, and one branch goes to AMP and the other branch goes to GMP, right? Each of these is inhibited by feedback inhibition by products of this reaction. So, for example, this pathway here is, before it branches at IMP, the enzyme that first catalyzes this first reaction is blocked by feedback inhibition by IMP, right? The basically the product of this first part of the pathway, right? But it's also blocked by the product, the second part of the pathway, which is AMP and GMP. So there's feedback inhibition, allosteric inhibition, that blocks this enzyme's function, right? The enzymes that cause the branch from IMP to GMP are also blocked by the product of the branches. So GMP here feeds back to inhibit the enzyme that does this reaction right here, but not the one that goes towards AMP, right? This is regulated independently. AMP inhibits its own synthesis. GMP inhibits its own synthesis, right? They can both feed back to inhibit the whole pathway if there's too much of this made, right? Okay, get the idea? Okay, of course, drugs used to fight diseases. Remdesivir is a small molecule. Here it is, and it is causing the inhibition of an enzyme called replicase, or RNA-dependent RNA polymerase, or replicase function. So this is an enzyme needed to make an RNA copy. It's actually right here, and this enzyme this enzyme does not function very well in the presence of replicase. I think in remdesivir, I think, blocks the active site of this enzyme. So it's basically a competitive inhibitor. And when remdesivir is present, the enzyme doesn't function. The virus can't make copies of its genome, and it cannot make copies of itself. So remdesivir is actually an fairly effective anti-SARS-CoV-2 drug. It's also very expensive. All right, so we have other examples of allosteric inhibition in long pathways. I don't need to go through all these, right? And I think I showed most of you guys the biochemical pathways chart of all the pathways in a cell. They're all have feedback inhibition and stuff involved, right? So you can click on this link to look at this when you look at the PowerPoint. a separate time. All right, that does that. That's enough of that. So we're done. Thank you.